onion and cheek cells. The cell is the basic structural and functional unit of all known living organisms. Some organisms, such as bacteria, are unicellular, consisting of a single cell. Other organisms, such as humans, are multicellular, having many cells. A cell mainly consists of a fluid-filled space called cytoplasm and is surrounded by a cell membrane. The cytoplasm contains a nucleus, mitochondria, Golgi bodies, lysosomes, endoplasmic reticulum, ribosomes, and vacuoles. Plants have an additional boundary wall called the cell wall, which is absent in animal cells. Plant cells have large liquid-filled vacuoles, which occupy most of the volume in the mature plant cell. Plant cells also contain chloroplast, which is absent in animals. To prepare a stained temporary mount of an onion peel. To do the experiment, we require cover slip, glycerine, glass slides, onion, watch glasses, distilled water, saffron in solution, forceps, brush, needle, compound microscope, blotting paper, and a dropper. Procedure: pour some distilled water from a bottle into a watch glass. Take half a piece of onion and remove leaves from the onion piece. Remove a piece of transparent onion peel with the forceps. Put the epidermis into a watch glass containing distilled water. Take some saffron in solution using a dropper. Put a few drops of saffron in solution into another watch glass. Using a brush, transfer the peel into the watch glass containing saffron in solution. Keep the epidermis for 30 seconds in saffron in solution so the peel can become stained. Take the peel from the saffron in solution using the brush and put it in the watch glass containing distilled water. Using a dropper, take some glycerine and put 2 to 3 drops of glycerine in the middle of a dry glass slide. Transfer the peel to the slide containing glycerine using the brush. Take a cover slip and place it gently on the peel with the help of a needle. Remove the extra glycerine using a blotting paper. Take the glass slide and place it under the compound microscope. View the slide under the compound microscope. Observation: a large number of regularly shaped cells are lying side by side. Each cell has a distinct cell wall. A distinct nucleus is present on the periphery of each cell. Lightly stained cytoplasm is observed in each cell. A large vacuole is present in the center of each cell, surrounded by the cytoplasm. Conclusion: as cell walls and large vacuoles are clearly observed in all cells, the cells in the observations are plant cells. Precaution: use a brush to transfer peel from one apparatus to another. Staining of the peel should neither be too dark nor too light. Extra glycerine stain should be removed by using blotting paper. To prepare a stained temporary mount of human cheek cells. To do the experiment, we require toothpick, cover slip, methylene blue solution, glycerine, glass slide, distilled water, brush, needle, compound microscope, filter paper, and a dropper. Procedure: pour some distilled water from a bottle in the middle of a dry glass slide. Take a clean toothpick and gently scrape the inner side of the cheek. Rub the cheek tissue on a glass slide that contains water. Mix the mixture using a needle and spread it. Take methylene blue solution using a dropper and add a few drops of it to the mixture on the slide. After 2 to 3 minutes, remove excess water and stain using a blotting paper. Take glycerine using a dropper and add a few drops of glycerine to the test mixture. Take a clean cover slip and lower it carefully on the mixture using a needle. Using a brush and needle, press the cover slip gently to spread the epithelial cells. Remove extra liquid around the cover slip using a blotting paper. Take the glass slide and place it under a compound microscope. View the slide under the compound microscope. Observation: a large number of flat and irregular shaped cells are observed. The cells do not have a cell wall. However, each cell has a thin cell membrane. A deeply stained nucleus is observed in the center of each cell. No prominent vacuole is observed in the cells. Conclusion: if the cells observed do not have cell walls or prominent vacuoles, the cells of the specimen on the slide are animal cells. Precaution: ensure toothpick used to scrape the cheek is clean so as to not cause infection to the cheek. Extra stain should be removed by using blotting paper.